I think I've mentioned this once before, and it may sound a little crass, but if you took dog poop, right? Dog feces, and you very carefully analyzed it, I'm sure you could find some molecules of glucose, some molecules of sugar in there, and you could find some lipids, and you could find some carbohydrates, some starches. Those three things are the ingredients of a birthday cake. But if you think natural processes are going to take dog feces and produce a birthday cake, nobody would ever suggest that, right? It makes no sense whatsoever. But that's what we're talking about here. This is exactly what these folks are suggesting when they're saying that these processes produce the seeds of life.
Hi, I'm here with my friend Rob Stadler. We've done a couple of videos with Rob before, and today Rob is going to be critiquing a paper that's recently come out. Really fine work, but then there was an application and a suggestion for origin of life, and he's going to be talking about that. Just so that you understand who Rob is, Rob Stadler earned a PhD in medical engineering from the Harvard-MIT Division of Health Sciences and Technology. As a scientist in the medical device industry for almost 30 years, he's obtained more than 175 U.S. patents and has more than 30 peer-reviewed publications, and has contributed to medical devices that are implanted in millions of patients worldwide. His experiences in gathering clinical evidence and in working with the FDA, that's the Food and Drug Administration, on the approval of medical devices, it's prompted him to write a book called The Scientific Approach to Evolution, What They Didn't Teach You in Biology. And that was the topic of our last conversation. But today we're going to be learning about a recent piece of work that has come out in the literature. And let me just say in the beginning that the senior author on that work is a friend of mine. His name is Richard Zare. And I have the highest respect for Richard Zare's work. I think for Richard, a Nobel Prize is way overdue for him. He does tremendous work. And what we're going to be looking at today is that work, but also the extrapolation and the suggestions of that work to be relevant to origin of life chemistry. So that's what we're going to be talking about today. And with that, I want to turn it over to you, Rob. Go ahead.
Thank you, Jim. It's a pleasure to be back here again. And today we're actually going to look at multiple situations, including that paper by Richard Zare, where natural processes do produce a lot of organic molecules. But a lot of the times when this is brought up, there's a lot of hype to it because they relate it to origin of life. And they don't talk about the reality, the fact that there's a diverse mixture of thousands of organic molecules, different types of molecules in there, that really make it impossible for anything to progress toward life. It's basically just organic garbage, as I call it here. So let's start with that paper that you mentioned here with Richard Zare as the senior author. And the title of the author, the title of the paper here, you can read yourself very clearly, but it's fascinating, a new type of chemistry, really, that microdroplets can force chemical bonds like carbon-nitrogen bonds that you might not expect you'd get just by spraying water into the air and creating little microdroplets. So that's all fascinating, and there may be some real important practical applications of this, like maybe you can make fertilizer for a lower price or greater quantity, or something great comes from it. But what concerns me is not at all the chemistry and not the claims about the chemistry. That's all fine. It's more about what they want to apply it to and what they're claiming in terms of origin of life.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together and I will share with you about why I embrace the resurrection of Jesus.
And I'll highlight here these you can see the highlighted lines here. So in this abstract, they're saying that they get similar findings to the Miller-Urey experiment, which I think most people are familiar with. It's now 73 years old. And that these observations provide a possible mechanism for making the building blocks of life on early Earth. That's the part of this that causes me a little bit of concern, not at all the chemistry that they're showing. Because when you make statements like this, the media engine starts to get hyped up and it starts to produce this hype about origin of life. And I think a lot of people are misled by hearing this hype that pummels us all the time. So here's one example. This is a Popular Mechanics article, and it's based on that paper, and it says tiny sparks in water may have triggered life on Earth, a new study suggests. And so it's kind of easy to blame the media because they're overhyping things. But in this article is this quote that microelectric discharges between oppositely charged water microdroplets make all the organic molecules observed previously in the Miller-Urey experiment. And we propose that this is a new mechanism for the prebiotic synthesis of molecules that constitute the building blocks of life. A lot of excitement there. And you might say, well, that's just the media hype, the media engine. But this quote actually came from Richard Zare, the senior author. So sometimes the authors themselves are responsible for some of that hype. And of course, this is picked up by many different outlets. Here's another Science Daily that micro lightning in water droplets may have sparked life on Earth, may have helped create the building blocks necessary for early life on the planet. And so let's look into the paper a little bit and just see what I'm talking about here as far as how this could relate to origin of life. So they have this apparatus, and they pull out all the room air from the apparatus, and then they spray water into the apparatus at high pressure with some nebulizing gas to make very fine water droplets. It's kind of like a high-tech sneeze, if you will, into the container. And the nebulizing gas includes nitrogen and methane, carbon dioxide, and you get these little micro droplets. And what's interesting is they have electric charge on them, some of them positive, some negative. And as they begin to interact, you can even get, as they show in this manuscript, you get some little micro light emission. So there's enough energy there to emit some light. They call it micro lightning. But you can see some of the chemical reactions that this results in here. And then they have the mass spectrometer here at the bottom, which is able to detect what kind of chemical reaction, what they produced, right? And that's where it gets interesting.
So let me if I could just mention a couple of things on this. This idea of getting these droplets with these charges and then getting reaction chemistry to occur, it's very interesting. In fact, in my own lab, we have been noticing what happens when you apply an electric field across molecules and across a material, and how it lowers the activation energy to convert those materials into a product. It actually cuts the activation energy in half, and that's what we're measuring. And it's interesting, even the way that Professor Zare describes this. He says that you're creating these charges on these atoms, and then so you're drawing out electrons, which makes them a lot more reactive. And so the way we have described it is actually a very similar way. We say we go from a closed shell atoms to open shell atoms, which is saying exactly what he said, and that then makes them far more reactive. So the chemistry here I find fascinating, and well worth disclosing in this fine journal where this Science Advances, where this is published. And it's interesting that he's able to get all of this chemistry happening. There's an enormous amount of chemistry that is going on. An enormous amount of molecules can form in this system, which then is part of the problem when it comes to origin of life, as you're going to talk about. But then let me also mention that he has the mass spec detector, and mass spec is a very powerful tool in being able to detect very small quantities of compounds, even down to the few molecule level. You can detect them. So the molecules don't have to be. It can detect things in quantities that would never be synthetically useful, but it can detect them. So he's got a very interesting proposal here as to how these molecules are forming. He's got a very interesting technique. Whereas in Miller-Urey, they used applied a high voltage to simulate lightning strikes. Here, what Professor Zaire is showing is you don't even need that. You can just generate these particles in a nebulizer, which it would be similar to, say, an ocean mist or something like that, and that you can get these charges. These charges are then causing these reactions to occur through this opening of the shell and through these pulling out of electrons, and now they become far more active, and you get this mixture. And I think what you're going to be getting at, Rob, is the problem is in the mixture of compounds that you get, the diversity of those compounds, and probably the yields of these as well.
Yeah, and you can see from what is shown here that this article, this manuscript clearly focuses on the molecules of the origin of life. They're showing amino acids and they're showing nucleobases. That's of interest to origin of life. But what they're conveniently not showing here is all the other stuff that's going to be produced. And when they actually show the mass spec results, this is taken right out of the paper, and they show the molecules of interest for life, glycine, amino acid, and uracil is a nucleobase for RNA. And I have no doubt that they've made these molecules and they're showing them, and that's just fine. But to show spectra that is this narrow in scope, this is going from 61.038 to 61.042, and I think they're doing that just because they want to show this urea finding. But what they're not showing is the entire mass spectrum, which shows, I'm certain it would show thousands of different compounds like this, thousands of different molecules, and just a few cherry-picked ones here are of interest to life. And that's why they're showing that. So I kind of wonder, why aren't they showing all the other stuff that was produced, or even mentioning it as a limitation, because it's a major limitation for trying to get life started if you have a bucket full of garbage. That's the point.
I have long talked about this in my lectures. I've long talked about this, that these mixtures are not useful for doing synthesis. It's very easy to detect very small quantities of molecules. One has to be concerned about the mixtures, and this is what Rob is getting at. These sorts of mixtures are not useful to us, and when you show a mass spectral region like this, yes, it focuses on what you want to focus on. But there's undoubtedly many other amines in there, many other carboxylic acids that would all compete for these reactive sites, for these subsequent reactions. So it's the application, the suggestion of the application of this to origin of life that makes it difficult to fathom when you get mixtures like this. Is that right, Rob?
Yeah, and I would love to see what you just said stated as a limitation in the paper, or as a just thinking into the next steps of where these products would go next. That's when this comes up. And this paper, of course, is just one in a slew, as I said, a slew of opportunities to produce organic molecules that end up being just basically piles of organic garbage. Here's another one. You're probably familiar with the Murchison meteorite. It fell in Australia many years ago. But just in 2008, they did some analysis on it, very similar to what we just showed with mass spec. And they find organic molecules related to life, like amino acids, and off goes the hype machine. And so here's a Science Daily article, "Life's raw materials may have come from the stars." And this is from the author of the manuscript there in 2008. "We believe early life may have adopted nucleobases from meteoric fragments for use in genetic coding, which enabled them to pass on their successful features to subsequent generations." That's a pretty big stretch from having found a few amino acids hiding inside of a meteorite. And we're going to explain why. But here's some more hype from this.
Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved.
That's right.
That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org. Thank you so much.
BBC Science Focus. We finally know how life on Earth started. So there you are, Jim. We finally know how life on Earth started.
Well, Rob, welcome to my world. I mean, this is what I got to deal with, and this is what's so confused the general public. This is a big problem. This is why when we've taken surveys, 2/3 of the general public thinks that scientists have made life in the lab. And so it's because of articles like this, and we are so far from that.
Yeah, and I just think it's misleading, and I don't know why we can't just try to have the responsibility of having the public know and appreciate reality here. A little background on this. This came from asteroid Bennu. I think you should all be familiar with that. It's all recent findings. So NASA had this OSIRIS-REx mission, which is super cool, to go up there and grab a piece of an asteroid and bring it home. It's all fantastic, and I love it. But the problem is, when I knew they were bringing this sample back home, in my head I'm thinking, oh great, here it comes. We're gonna have hype like this being generated from this thing, instead of focusing on just the facts, just the chemistry. So we have people being misled with statements like this. And here's another one, also from asteroid Bennu. The historic asteroid sample reveals the building blocks of life are, in fact, extraterrestrial in origin. So,
which is actually just the opposite of what Professor Zaire is saying.
Yeah, he's saying they come from
from these droplets,
crashing.
and this one is saying no, they come from outside the Earth.
Yep, a little disturbing. And here's a couple of quotes, and these quotes come from the senior author from this manuscript that it came from. They did the analysis on the sample from asteroid Bennu. This is all very exciting because it suggests that asteroids like Bennu once acted like giant chemical factories in space and could have also delivered the raw ingredients for life to Earth and other bodies in our solar system. So it's kind of creating this hype. And then this next quote, very interesting. And there is also the puzzle of why life didn't form inside Bennu itself, given that most of the raw ingredients needed were present.
Oh, that's pretty bad. That's really pretty bad. You know,
well, it's a puzzle. Why didn't life form inside the asteroid itself?
It's not a puzzle. Not a puzzle.
I don't think there's any puzzle there, but
there's no puzzle.
It scares me that people are presenting this. All right, here's another one from asteroid Bennu. This actually, I put this in here because look at the date. Yesterday. Okay, this came out yesterday. So this stuff comes out constantly. It's just flooding our culture. So it blew us away how an asteroid may have delivered the vital ingredients for life on Earth. There were things inside the Bennu samples that completely blew us away. That's fine. You know, you're surprised at what you found on there. That's fine. But it's how you interpret it. These strongly suggest that asteroids similar to Bennu crashed on Earth, bringing crucial ingredients that led to the appearance of life here. There you have it. So I hope you're getting a feel for this hype, and it's very prevalent, and we're getting blasted by it all the time. Here's another one. This is a Japanese satellite that found two dense icy objects in space, you know, and from the light coming off of them, reflected off of them, they found it contained organic compounds, and they hyped up there. The hype machine is running, and it says the mystery interstellar icy objects are carrying the seeds of life. So there's plenty of it out there. Mm-hmm. And so all of this comes together to tell us that, yes, in fact, there are many different ways where natural processes can produce diverse mixtures of organic molecules. We've got all the Miller-Urey, the microdroplets, asteroids, all this stuff. There's even, you know, hype of exoplanets now and being able to find organic molecules on exoplanets. And we're not at all doubting that these organic molecules are out there. No problems with that. It's more about, so what? What does this have to do with origin of life? And you're probably wondering why I keep calling this organic garbage. And let's get into that now. Let's switch topics to get into why do I call this organic garbage? What else is in there is the question. And here's some great quotes. You have to dig into the literature, though, to find these things, because these quotes are kind of hiding amongst the hype. And so for the Murchison meteorite that I mentioned, there's this great quote. The Murchison meteorite contained tens of thousands of different molecular compositions and likely millions of diverse structures, which suggests that the extraterrestrial chemodiversity is high compared to terrestrial relevant biological and biogeochemical driven chemical space. That's a lot of complicated words, but it's basically just saying there is so many different kinds of molecules in there. It's just a pile of organic garbage. And if you search very hard and long, you can find amino acids, right? And you can find your nucleobase and write a paper about it. But it's useless because it's caught up into this pile of garbage. Looking at the Bennu, all the hype from asteroid Bennu, the actual paper in Nature Astronomy, it says the mass spectra of the extracts contained tens of thousands of compounds with mass to charge ratios between 100 and 700 that correspond to about 16,000 molecular formulae. Okay, so they're admitting this, but they're kind of hiding it in the background and hyping up the fact that they found some amino acids. And then here's some more. And I really applaud here Jack Szostak, and you have to search hard for this, but he admitted the problem that Jim and I are talking to here, which is, he says in this video, you can look it up yourself, he says here, the problem is that those collections of molecules are mostly things we don't want and very little of the right molecules in very, very low concentrations.
Now that's, I mean, good for Jack to have said that. Most origin of life researchers don't say that. The only other person that I know that says something like that is John Sutherland. But yeah, that is the problem. That's the key problem. So we're not the only ones saying this. This is Jack Szostak, a major origin of life researcher, and he's a Nobel Prize winner. And that's the cold hard facts on this.
Yeah, and if people want to take up issue with this video and nitpick what we're saying here, I think you're going to have to take it up with Jack Szostak and John Sutherland too, because they're on our side. And I can ask you, Jim, as a synthetic chemist, your whole life you've been doing this. I mean, what happens if you start with very little of the right molecules in very, very low concentrations and you try to accomplish something? You know?
Yeah, you can't do chemistry on any of these mixtures. So I have long said, has anyone been able to show doing chemistry on mixtures like this? Because what you can do is you can mimic this. In other words, you look at those mixtures, you look at the 16,000 compounds, and you could make that. You could add all the different types of compounds together and then try to do any chemistry on that. Or try to do a separation. We can't do the separations because many of those compounds have very similar structures. And when you have that many, it just can't be done. So you know, it's interesting because you can take a whole handful of supplements in the morning, vitamins and supplements, and your body knows exactly what to do with it. Biology is like that. Biology has enzymes which segregate this and puts it into different places. But pre-biology, meaning abiogenesis, before biology takes off, you don't have the compounds to do this. And there's no way that anybody can use these mixtures. Now let's take it another step, Rob. Even if you were presented with this, could you do anything? The answer is no. Now, but even if you were presented with these 16,000 compounds, or however much Professor Zare had, if you were presented with these in discrete bottles, separated, could you use them? And the answer is no. We don't know how to use them to make life. So you can take all the components of life and give it to a researcher, not on an early Earth, but in a modern laboratory. You can go even further. You can take a cell, A working cell, you can deconstruct it, take it apart, and put all the different pieces in known bottles, and even have all the polymers made so you already have the code. There is no scientist on Earth that would ever suggest that they could put that cell back together again. So we're so far from being able to build life that these are crazy suggestions, and it just is no wonder why the world is so confused. But go ahead, show more about what these guys are saying.
Yeah, and I mean, I kind of think that's the key message. It's basically irresponsible, I think, to get the public hyped up about these things. And I know people are curious about life on other planets and life on asteroids. Of course, they're curious, and they're kind of eager and hungry for that hype. But it's not good to feed into it, is my point. So similarly, Origin of Life chemist John Sutherland said, and Jim, this was in one of your talks last week too, but John Sutherland himself said, I don't think meteorites make the right sort of mixture. We need to have more constrained chemistry to actually make the right sort of mixture, right? So what he means is you gotta control the chemistry so you get only the things you want and you don't get a pile of garbage. It has to be something fundamentally different to give you what you want in decent concentration, right, and purity. That's the problem. And so, these things are actually, what they make, this pile of garbage, is so complicated that the mass spectra becomes so complicated, it's very hard to even tease everything apart. Who wants to go after looking at 16,000 different components? And so a way to approach that is through computer simulation. And this is a paper in Science where they did a computer simulation of very simple molecules advancing toward what they said is, in the paper, advancing toward prebiotic chemistry. And so in this computer simulation, they started with just 6 pure ingredients. So this is pretty similar to the Miller-Urey experiment. And they conducted just 7 rounds of chemical reactions between these reagents. And what they end up with, because it's a computer simulation, you can very clearly count and itemize everything. And they produce 36,603 different molecules that are not associated with life, and only 82 different molecules that are associated with life, right? So amino acids, maybe nucleobases. So you have this ratio of 82 to 60, 82 desirable versus 36,000 junk. And it's not just junk, it could be toxic. It's going to interfere with whatever reactions might happen next. So you have 99.776% junk. And how are you ever going to get out of that something useful, is the point. And I think I've mentioned this once before, and it may sound a little crass, but here's a very good analogy to this. I think is really accurate. But if you took dog poop, right? Dog feces, and you very carefully analyzed it, I'm sure you could find some molecules of glucose, some molecules of sugar in there, and you could find some lipids, and you could find some carbohydrates, some starches. Those 3 things are the ingredients of a birthday cake. But if you think natural processes are going to take dog feces and produce a birthday cake, nobody would ever suggest that, right? It makes no sense whatsoever. But that's what we're talking about here. This is exactly what these folks are suggesting when they're saying that these processes produce the seeds of life. Any comments on that, Jim?
Yeah, well, and all of what Professor Zare made, and all of what you find there, that's all racemic. So even when you have those compounds, you have the mirror image of them. So he shows glycine, but that doesn't have a stereocenter. But when things start having stereocenters, you have the enantiomers of them, and when they start having multiple stereocenters, you have the diastereomers. So that it becomes even more confounding of how you would ever use those and separate those. So it's just a big problem. So this just goes along with what we've been saying, that these articles come out. The origin of the researchers will sometimes make claims that go way beyond what the data suggests, in their extrapolation when it has to do with origin of life. I'll tell you that Zare paper has enough meat in it to be a very interesting paper without the suggestion that this somehow bespeaks the origin of life and the molecules that go along with the origin of life, because all the molecules that they made would be useless in the form in which they were made. And that's the problem with it. And so it just brings it a little bit too far. And a lot of times scientists make extrapolations, and those extrapolations are fairly innocuous. They don't affect the world. Nobody takes those and runs with it. But in this case, it's gotten so far out of hand that we need to put some constraints on this and say, hey, let's be more circumspect before we make these. Or if we're going to say that, yes, we see these molecules that are the building blocks of life, put the qualifying statement, but in these cases, this would not be usable molecules because of the very low yield and the great diversity of compounds that are there. And when you have compounds that I'm sure there's lots of other amines, lots of other carboxylic acids that would all compete for these active sites and shut down anything. And when you have a mixture of 60,000 compounds or 36,000 compounds, that's not a usable mixture. Even a mixture of 5 compounds can often be hard to use when they have related structures. So yeah, that's the problem from a synthetic chemistry standpoint.
So if you had a reaction vessel like Richard Zare had, and you did his process, and out came amino acids in what kind of purity are you looking for? You're looking for 95% purity, something like that, to get excited?
If you're going to use it directly, I mean, if I had 95% purity, I would bring it up to much higher than that before I went on to the next step, so that it wouldn't interfere. But the problem, if you have 95% purity, you can probably purify it up to 99.99% purity. But if you have something at less than 1% purity, it's very hard to bring it up. There's just too many confounding molecules in there. That's the problem. So it wouldn't be useful. And any organic chemist sees what I see. This would be very clear to them.
Yeah, as you said, I'm grateful to John Sutherland and to Jack Shostak for agreeing with us. Although they haven't seen this video and agreed, I think they would. I believe they would.
Mm-hmm. Yeah, I think so too, and I think that Professor Zare would agree with it as well. That probably the suggestion that he made in the literature, both in the literature and then also to that reporter, probably went a little bit too far. And like I said, I mean, hyping work is part of the salesmanship or the salespersonship that's done in trying to get attention for your work. But in this area, it's so confused the general public that it starts having a real effect. And it's these sorts of articles that generate the data that gets into our textbooks. And that's what makes it very confusing. All it would take is a few more sentences to say, look, we've got these mixtures, it's got these compounds that are for life, but it's got 1,000 times more compounds that have nothing to do with life, and therefore that mixture would be irrelevant for moving on toward life based on what we know today. Maybe we could be surprised by something in the future, but for right now, these mixtures are irrelevant. So to suggest that those were the compounds that sparked life, we have no way to suggest that based on what we know today. And so somehow writing a few sentences of qualification would probably help. And when speaking to the press, to speak in a guarded fashion, because the area has gotten so overblown. That's the problem with it. Although the science of what he was doing was super interesting, in my opinion, as a chemist. Yeah. But it was just the extrapolations that went too far.
Yeah, I completely agree. Just 2 sentences to expose your limitations. That's kind of all we're asking for.
Yeah, and that's something that you talked about in your last video that is required when you are trying to get something through the FDA and approval, that you have to show the limitations. And if we just walked in that a little bit, that would probably go a long way here.
Yeah, and I wanted to mention that. When I write a manuscript in medical literature, you really have to have a section of the discussion that's called limitations, where you openly disclose, you say, hey, we only studied women, and so our result may not apply to men, right? That's a limitation. Simple things like that are required, and it doesn't seem to be generic. It doesn't seem to be generally required in all science literature, and I think it should. Mm-hmm. So we're kind of wrapping up here, but I just wanted to mention that this is something that's been known for a long, long time, what we've been talking about. In fact, it has a term to it. It's called the asphalt paradox of the origin of life. And here's a quote from this paper. An enormous amount of empirical data have established, as a rule, that organic systems, given energy and left to themselves, devolve to give uselessly complex mixtures, asphalts. Further, chemical theories, including the second law of thermodynamics, bonding theory that describes the space accessible to sets of atoms, and structure theory requiring that replication systems occupy only tiny fractions of that space, suggest that it is impossible for any non-living chemical system to escape devolution to enter the Darwinian world of the living. So I've been calling it organic garbage. You could also call it asphalt. You could also call it tar. It's all kind of similar, but it's just a random pile of very complicated chemical mixture that is basically useless.
Yeah, and Steve Benner talks about this as well, the asphalt paradox.
Yep, I think he's on this paper. I just didn't list him here.
Yeah, because I think that Dr. Kim, who's listed there, is his coworker. So that's the problem. And Steve Benner, an organic chemist, he sees the same thing. You have to have, as John Sutherland says, you have to have more constrained chemistry going on.
And I think
this wouldn't do it.
That's quite an understatement. More constrained chemistry. I mean, like you said, even if you get to 95% purity, that's still on the edge of not being all that useful.
It's not useful. I mean, because what you have to do is you have to do what's called condensation polymerizations. And to do a condensation polymerization, you've got to have 99.99% purity in order to do a condensation polymerization. So 95% might give you like a trimer or a tetramer according to the Carruthers equation. So those just don't do.
And it's not just about the purity, right? We haven't talked about the mass transfer problem. And maybe you could add that to what you just discussed.
Yeah, the mass transfer problem, you have to, how much material? So say you had all the 20 canonical amino acids. Say you had the basic sugars, the 5-membered ring sugars, the 6-member ring sugars, the fructoses, and the 6-member rings, the glucoses. Say you had that, mannose, and all of these compounds that you need. How much would you need to bring these on to a cell? Because you're going to need thousands of chemical steps going on. How much would you need? You need lots of material. Because what happens is if your yield is, say, 50%, and 50% would be a pretty decent yield when there's a mindless early Earth that doesn't know what it's going toward. So you have 0.5 times 0.5 times 0.5, each one of these reactions being 50%, you run out of starting material. How do you bring up more starting material from the rear when you never kept a laboratory notebook, because nature never does that? That's the mass transfer problem. And how much material would you need even if you were a skilled chemist? To go through thousands of steps, you'd need tons of pure material. That's the mass transfer problem. So that gets you as well.
Okay, so just to bring this to a conclusion then, our point here is that many natural processes produce diverse mixtures of organic garbage. And the point is, so what? They have no opportunity to contribute to the formation of life. And we've only been talking about simple molecules so far. As the molecules grow more complex, the available chemical space that they can occupy, there's so many different varieties, it grows explosively. It's not just exponentially. So if you were to make molecules 10 times the complexity of what we've described so far, the opportunity to make the wrong molecule just grows exponentially, and you end up with even worse than what we have here. And a lot of these molecules can be toxic, not just in the way.
Yes, that's right. Yes, when you get to more complex molecules, you get numerous stereogenic centers. Those are what some people used to call chiral centers. And then you get what's called diastereomers, and then the mixtures are gross. I mean, just very hard to separate and very hard to deal with. And so it just gets more complex. So actually what we've been talking about is the simple cases. The hard cases are much worse than this. And you look at it, there's tens of thousands of compounds that are in there. And that's only the ones that are small molecules. You get lots of polymeric compounds as well, which gum up the works and make separations more difficult. So try to do recrystallization. Try to do crystallizations from those sorts of mixtures and get pure compounds. It's very, very difficult. And then when you have diastereomers in there, it gets even more difficult. Rob, thank you for helping us to work our way through this paper and to see that what we're saying here, this is routine, where people are coming with these sorts of mixtures and these sorts of problems. And all of these can harken back to the real problems of origin of life. But hopefully these will get, people will start seeing more of these problems. So really what we're going to need to solve origin of life is some dramatically new ideas, new ways that we could think about for how life might have begun, because drastically new ideas. And there are people trying to think of new ideas, and I'm trying to critique some of those ideas, and we'll take a look at those in the not too distant future. Thanks so much, Rob, for joining us. God bless you, my friend.
Thank you, Jim. It's a pleasure to be here, and God bless you. Thanks.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.